Hi class, and welcome to your next video on genetics. We're going to talk a lot about Punnett squares now, and we're going to talk about how you get your genes now that we've finished talking about DNA and what those genes do for us. So let's talk about first, where did this begin? Well, this began with Gregor Mendel, a monk from the Czech Republic. He's responsible for what is known as Mendelian genetics. And that basically means something that is dominant or it's recessive. It can't be any kind of mixture. It can't be any other type of thing. Only dominant or recessive genes, which is what we're going to talk about in this video. Mendel did a lot of experiments in the monastery, and he used peas to determine how traits were passed from generation to generation because he could easily manipulate those traits. Between the time and of the generations is fairly short because of their peas, so it was just literally weeks to months, and that made it very easy. So when we consider Mendelian genetics, we want to consider a couple things. Each trait is composed of two genes, one from mom and one from dad, so you're still always getting two forms of every trait. And parents can only donate genes that they have. So if they don't have a certain gene, they can't pass it on. So if a parent doesn't have a gene to be tall, if both parents are short, they cannot pass on a gene to have a tall child. Now, if they have a hidden gene, or what is called a recessive gene, they could pass that on. The physical characteristics, or how the trait presents itself, is known as the phenotype. It's how it kind of looks. Sometimes it might be, do they have a disease or not have a disease? Do they carry a certain protein? But it's how does it physically present itself. The actual genes that one has is the genotype, and a lot of times we're going to write these in the form of letters. They're going to be upper and lowercase letters. Sometimes they're going to have superscripts or subscripts, but they're really generally going to be letters. So what did Mendel study? Mendel studied peas, like we said, and they were strictly dominant or recessive. And you might be wondering, okay, you keep saying this dominant or recessive. Well, in peas, one dominant allele or gene is present. The organism has a trait. This is the same with you. If you have one dominant allele, you have the trait. You must have two forms of the recessive trait to have the phenotype of the recessive. So remember we said you get one from mom and you get one from dad. Well, if mom gives you a recessive and dad gives you a dominant, you're going to show the dominant form. Now if you get two forms of the recessive, one recessive form from mom and one recessive form from dad, you're going to show the recessive form. If the other case, you get two dominant forms, one dominant from mom and one dominant from dad, you're going to show the dominant form. So the case is, with a dominant allele, you must only have one dominant allele to present that trait. But with the recessive form, you must have two recessive forms. So we also look at generations with genes. So we start out at the parent generation, the P generation, and we go to the F generation. F actually stands for filial, F-I-L-I-A-L. And when they pollinate each other, then they go to the F2 generation and the F3 and so on and so forth. And we look at who has different traits and how they're being passed. And that's exactly what Mendel did. He said, okay, this pea plant is purple and this pea plant is white, and if I cross them, I'm going to get all white plants. And then he kept looking and looking, and he kept seeing different types of things happening. It wasn't the same thing over and over again, and that's where we got our study of genetics from. So Mendel has two laws based on his work that are able to help explain inheritance, and you've already heard of one. The law of independent assortment we've already kind of talked about, And the law of segregation is the second one. So the law of segregation, two members of a gene pair or allele segregate from each other in the formation of gametes. So remember, gametes are sex cells, and half of the gametes carry one allele and the other half carry the other allele. Only one allele is placed in each gamete. So then we can only donate one allele to the new organism, but the other parent is going to donate the other allele. This happens during the process of meiosis when we are forming those gametes. The law of independent assortment then states that allele pairs separate independently during the formation of gametes. This means that traits are transmitted to offspring independently of one another. So traits do not necessarily follow each other. For example, blonde hair and blue eyes do not go hand in hand with one gamete. So as long as they are not on the same chromosome, they can assort independently of each other. It does not matter where they are. So the Punnett square. Punnett square is a diagram that is used to predict the outcome of a certain cross of two organisms. And as we can see, we have two organisms that we're going to call heterozygous. And we're going to call them heterozygous because they have a big R and a little r. And all capitals are used to represent dominant alleles. And all lowercases are used to represent our recessive alleles. And when we have a dominant and a recessive together, they're going to be what is called heterozygous. When we have two dominants or two recessives, they're going to be what's called homozygous. And what we do is we cross them together. And as you can see, we're going to just draw lines in order to see where the actual alleles go. And I'm going to show you this in just a second. So as you can see here, and let me get my ink here. What we're doing, if we come down here, we're bringing this down, and we're bringing this over. And we're doing the same thing. Bring this down, and we're going to bring this one all the way over. Bring this one down. Bring this one over. Bring this one down. Bring this one over. And we're just kind of matching them up in order to see what would be the outcome of this cross. So the terms for describing these genes, like I already told you, homozygous genotype, which is also known as true breeding, is when the genotype has two of the same trait. It can either be homozygous dominant or it can be homozygous recessive. Remember I said that anything that is dominant has capital letters and anything that is recessive has lowercase letters. It just has to have two of the same, so it has to have two capital or two lowercase. A heterozygous genotype, which is also known as a hybrid, is a genotype with one dominant and one recessive allele. It can only be one capital and one lowercase. So let's look at this. Dominant genes. Dominant genes are the preferential genes showing itself if present at all. We only need one, and they are signified by a capital letter. So which of the following are going to have a dominant allele? For number 3, number 4, which of the following are going to have a dominant phenotype? And write those down, and then I'm going to go ahead and circle the ones that are correct. Now, if you wrote down the ones that are circled, or you circled them, wrote down all of them, and then circled them, those are the correct ones. So you got those right. So you are understanding what a dominant gene is. So let's go on to recessive genes. So recessive alleles can be masked if there is a dominant allele present. So you must have 2 in order to see the recessive phenotype. So which of the following have a recessive allele? What this question is asking, do you have one recessive allele here? Which of the following have a recessive phenotype then? So remember, you need 2 to have a recessive phenotype, and go ahead and write those down. Now, if this is what you had written down, then you were correct. Remember with the recessive allele, number 3, that question, you only need 1 to have a recessive allele, but you need 2 for number 4 to have the recessive phenotype. So practice problem 1. In tomatoes, red fruit is dominant to yellow fruit. Cross a homozygous dominant, which means capital R, and a homozygous recessive fruit. So, what is your P generation? So write that down, and I'm going to bring it back. Okay, so your P generation is just your homozygous dominant by your homozygous recessive. So you should have capital R by your lower R, just like I have here. So B, what is the genotype of the F1 generation? So we're going to need to do a Punnett square. So you're going to need to do a Punnett square of the capital R by the lower R. So go ahead and do that, and then write down, you should actually only have 1 answer for the genotype of the F1 generation. So go ahead and do that. So you should have crossed that, and you should have seen that you would only get a capital R, or a heterozygous genotype, which is also known as a hybrid. So what this is asking next on C, what are the possible genotypes of your F2 generation? So you need to take and cross the 2 of the heterozygous genotypes together to see what you get for your F2 generation. So what I want you to do is to do a cross for this, do a Punnett square, and write the number of each different type that you get. So what you should have gotten with your Punnett square was what we call a 1 to 2 to 1 ratio. 1 heterozygous dominant, capital R, by 1 homozygous dominant, capital R, by 2 heterozygous, by 1 homozygous recessive. So the next one you're going to do is, in humans, brown eyes are dominant to blue eyes. A man who is heterozygous for brown eyes marries a woman who is heterozygous for brown eyes. Can their children have blue eyes? So, what do you think? If you said yes, you are correct. So you should have probably done a Punnett square to think about this. So it would be like capital B lowercase b by capital B lowercase b. And when you do that, you get that 1 to 2 to 1 ratio, and you have a 25% chance of having a baby with blue eyes. So they have 25% chance for B. Okay, this is the end. So we'll talk about this a little bit more in class.